from Virginia Tech, and she's in the crop soil environmental sciences department. And in that department, it's similar to our undergrad plant science program in which you have specializations. And in her specialization is plant breeding. So she's going to talk more about what her PhD topic is and her educational background. What I wanted to really highlight about Bodie, which is something that our committee sort of recognized, was at Virginia Tech, they have what they call a graduate teaching scholarship program. So this is a competitive program that graduate students across their College of Agriculture can apply for. And she was selected, and it really is designed for students who are interested either in a high percentage of a tenure track position where they have a high percentage of teaching, or a lecturer position where you're 100% teaching. So not only are you doing your dissertation and your area of discipline in science, you're also getting mentoring and learning about pedagogy and really getting training on how to teach in a higher education academic setting throughout your PhD program, which to me is just a phenomenal, learning about it is just kind of a phenomenal experience for a graduate student. So as part of that program, she actually is an instructor of record. It's kind of crazy right now in plant breeding and genetics. So it's a 4,000 level course. I asked her, like, what did you have to do to get here? Because they're still in session just like we are. So somehow, miraculously, not knowing that she was going to get an interview, there were some field trips planned for this week. So she was able to kind of choose a week that worked out well for her. And then additionally, besides that level of anxiety and stress, right before she got on the plane, like the night before, she had to submit her dissertation to her committee. And when she gets back, she has the pleasure of her defense. So it's a lot for a person. We really appreciate you taking your time out of this, your schedule. It's a really busy time, and just want to acknowledge, it's been really a pleasure for you to have you here. Meeting with faculty and just learning a lot more about you. So thank you for taking the time out of your schedule and applying for the position.
Thank you. Well, thank you, Rose, for that great introduction. And I've really enjoyed getting to know some of you over the last couple of days. I'm really impressed with this turnout. I'm really flattered you all have taken the time to join me today. And as Rose mentioned, so I'm at Virginia Tech. I'm from Virginia originally. So I'm from a little town called Smithfield. If some of you partake in Smithfield ham and bacon, that's where it's from, ham capital of the world. We're also just about 20 minutes down the road from where the first peanuts in the US were grown, so very proud of that. And before I go any farther, I have brought some Virginia peanuts for you all to enjoy. Does anyone have an allergy before I pass them out? They're in little individual snack packets. Nope, great. Well, if we want to start passing these around, I can do that. Thank you. And that'll tie into the lecture too. It wasn't just a bribe, but I'm very proud of being from Virginia. So for my education, I started at Virginia Tech. I started in basic biology major. I was thinking pre-med, quickly realized I did not have the stomach for it. And just by chance heard a guest lecture on plant breeding. Called at College of Agriculture that day, quickly transferred in, and I found my people. And it's the people that have really kept me in agriculture. So after finishing my bachelor's, which was crop and soil with a focus in plant breeding and genetics, I went on in a roundabout way to finish my master's at NC State. So I started at Clemson. My advisor took a position at NC State halfway through my master's, so I transferred to NC State to finish under him. Which was a great opportunity because then I got to see some of USDA plant breeding research as well. So I focused on soybean for my master's. And then once I completed my master's, I had the opportunity to return to Virginia Tech and pursue a PhD in soybean breeding there as well with Dr. John, soybean breeder. And so that's where I've been the last 3 years. And for my master's, since I stayed in the same crop, my master's research was focused on improving soybean tolerance to drought and flood stress. And then my PhD work has been more focused on innovations in the soybean feed and food industries. So how can we simplify processes of quantifying seed quality for farmers and producers, as well as improving some of our food lines like natto and tofu, which we are world renowned for. And one of our tofu lines won the national tofu competition in Japan a couple of years ago. So that's definitely a point of pride for our program. And as Rose mentioned, so in addition to my PhD research, I, after my first year, applied for and was accepted into this graduate teaching scholar program. It's a very unique program at Virginia Tech. And its mission is to develop inspiring and well-rounded educators, mentors, and leaders. And so, as Rose mentioned, this is a great opportunity for us to engage with teaching mentors, something that I feel like a lot of us don't get at the normal graduate level. And so I've been allowed to be the co-instructor and an instructor of record. And as Rose mentioned, timing lined up great. My students are on field trips. Haven't gotten any emails from the TA, so I think everything's going well. And they'll be learning from faculty at Virginia Tech this week in their different reading programs. And it was really this graduate teaching scholar program that made me realize I want to be with students in the classroom, in the field full time, and really dedicate my time to them instead of being a part research faculty. And so when this opportunity came up at UF, I thought that sounds perfect. Not only am I spending 100% of my time with students, but with UF being so focused in research, I can really keep my finger on the pulse of what's going on and present those new findings and new opportunities to students as we move forward. So that's enough about me. I want to know a little bit about you all. So this is a program called Mentimeter. If you have your phones and you'd like to participate, you can scan the QR code and it'll let you enter just one or two word response. And this is a program I like to use in class to get to know students. I also find for students who are a little more shy and hesitant, it allows them to answer questions and ask questions a little more confidently. Right, the QR code could have blocked some of the answers, but I'm seeing a lot of Brazil, Oklahoma, Alabama, Palestine, China, Mexico. Really a great representation. So thank you all for answering that. And then I have one more question for you before we get started. If it'll start. What do you love about UF? What brought you here? What keeps you here? What do you think is important for me to know as I'm making this decision? The weather is quickly growing. People was a strong second. That's great. I've got the research. I see students, your labs, all great things. I won't lie, weather would be wonderful. I'm tired of being cold. And I can see why the people have kept you here. I've met some fantastic people over the last couple days. And can see you all are a great community. Well, thank you so much for humoring me with those questions. And we can head into the lecture now. So I've designed this to be kind of an introductory lecture to maybe a couple lectures on photosynthesis or a module. And I think repetition is really the key, especially when you're talking about complex subjects. So I like to start big, set a firm or good foundation for students, and then really get more complex over multiple lectures or days. So that's how I've kind of designed this lecture today. And so we'll be looking at photosynthesis, light to life. And today what we'll be learning is the chemical reaction for photosynthesis. We'll talk about the different reactions, the light dependent and independent. And then we'll talk about the 3 pathways of photosynthesis and why understanding photosynthesis is so important. Now, I told you the snacks would tie into the lecture, so we'll be using a peanut plant as our model plant. And I want you all to think about as we go through all the processes that had to happen to take the energy from the sun for the plant to create the peanut that you all are enjoying today. So for photosynthesis, we need sunlight, our energy source. You need CO2 from the atmosphere. You need some water. And oxygen is produced as a waste project. I'm sure we're all very thankful of that. And then as well as sugar, glucose, carbohydrates, all refer to the same energy source that the plants use and that we enjoy when we consume them. And so from this, you get the completed reaction for photosynthesis. Again, you've got your different components, your carbon dioxide, your water, sunlight, and generates that sugar and your oxygen. But let's take a closer look at what's going on. So any of you who are familiar with Miss Frizzle and the Magic School Bus, we're going to kind of be doing that today. So let's look at what's going on with sunlight. Where is this energy coming from? So if we zoom in on a particle of light, principles of light. So a photon is the smallest possible particle of the electromagnetic energy. It's also the smallest possible particles of light. So this is what's traveling in the wavelengths, this is what's hitting the leaf. And the light, sunlight, has many different spectrums, but what the plant is utilizing is the visible light. And this is what we see with our eyes. This is what causes all the colors we see based on what's refracted, what's absorbed. And so the nanometers of this are usually from 400 to 700 nanometers wavelength. And this is what the plant is using. So if we zoom back out, we kind of have a better idea of what's going on with the sunlight now. But what's actually happening when that hits the leaf? So if we zoom in, here you can see the different cell structures. You've got the plant cell walls. You can see that green color that's coming from chlorophyll. And if we zoom in on a singular cell, we can now see the different organelles in it. So you've got your nucleus. We've got your mitochondria that they drilled into our minds in high school, the powerhouse of the cell. More specifically for humans and animals. You've got your vacuoles, which we'll discuss in a moment. But what we really want to focus on in terms of photosynthesis is your chloroplast, this structure up here. And so there's some interesting structures in there. So we're going to take an even closer look at what's going on in the chloroplast. And so within your chloroplast, you've got different structures. So you've got your stroma, which is kind of this fluid-filled space that surrounds the thylakoids. You've got your thylakoid here, which is this singular circle on top of this stack. And this is where all of the light dependent reactions are taking place and where the chlorophyll is located. And then you've got these stacks of thylakoids, or these granum. And you can see here kind of a cross section of what that granum looks like. It kind of weaves in and out, back and forth. You're increasing surface area. And does anyone have an idea of why increased surface area is important? If you have more area, is there more opportunity for light to hit it? Yes. So do you think that would increase or decrease energy production? Increase, right, yes. So that's why that folding is important. So now that we kind of understand the structure, let's look at what's going on in these structures. So as I mentioned, here you've kind of got your granum. And it's within this granum or these thylakoid structures where you have your light-dependent reactions, the ones that need the sunlight in order to work. So you've got your sunlight hitting it. What it's doing is it's splitting the water molecules into your oxygen waste, your hydrogen ions and electrons, and it's producing these energy molecules, ATP, and then NADPH, which will be used later for sugar production. So again, the light hits structures in the phylum, it excites the electrons, that splits your water molecule and generates those two proteins. Well, on top of your thylakoid, you've got these different pigment molecules, which are chlorophyll. And so you've got your light hitting these chlorophyll. That light is bouncing off of them, increasing. It eventually hits this special chlorophyll down here, which is where the electrons are split off. And it's where the electrons are used later for the energy production. And so this is on top of the thylakoids or on top of the membranes and called the antenna complex. So we kind of mentioned that chlorophyll are pigments, and we've kind of talked about light. And so both of these are really important because the pigments are responsible for the vast colors in plants, all material that you see. I see a lot of gray shirts out here, they're blue chairs, all of those have pigments. And as we mentioned, they're related to different wavelengths. So you can see here, I mentioned those special chlorophyll, they're at that bottom of that antenna complex. And what this is showing is that it's absorbing the blue and red light, so those 400 and 700 nanometer wavelengths, and it's reflecting the green, which is why we see it as green. And so it's these blue and red wavelengths that are really powering our photosynthesis here. So these are those pigment molecules that were on top of that antenna complex. And so if we look at the whole picture of what's going on in the thylakoid membrane, you can see here those antenna complexes that we just discussed. You've got your water being split off, electrons are being transferred over. Those hydrogen ions are hanging out on the interior of the thylakoids. And then we've got generation of NADPH and ATP. And what you might notice here, so one always comes before two, except in photosynthesis. They wanted to make all of our lives difficult. And what actually happened is that photosystem 1 was discovered and understood before photosystem 2, and they just kept that naming structure. So again, one before two, except for photosynthesis. So your first reaction in this light dependent is photosystem 2. You've got the electrons being transferred over an electron transport chain, photosystem 1. And so now we're going to take a closer look at what's actually going on here. So this is just another visual, another way to think about it. So in your photosystem 2, you've got your light energy that's hitting those pigment molecules. That's being excited, energy is being increased. It's hitting your special chlorophyll here, which in the case of photosystem 2 is P680. And so that 680 refers to the wavelength that's being absorbed. So it's absorbing 680 nanometer wavelength. So that energy has increased, it excites electrons. The electrons move up to a higher energy level, which then allows it to be transported on an electron transport chain across the membrane. And so then what's being generated, you've got those hydrogen ions we talked about that were being split from the water here. And then you've got your energy generation of ATP. So we're going to set ATP aside for a moment. So ATP has been created. It's waiting for us to use. Photosystem 1 has started. And so those same electrons that were first excited and moved across now have to be excited again in order to continue that transfer. So light is hitting these pigment molecules at that other antenna complex. Energy is being increased. The special chlorophyll in this case is P700. So what would the 700 represent? Wavelength, right. So you've got 680 in photosystem 2, 700 in photosystem 1. Those electrons are again being excited, so they're moving to a higher energy level. And that again powers that transfer over through the membrane on an electron transfer chain and generating NADPH. So now we've completed the light dependent reactions, the ones that directly require light for use. You've generated your ATP, your NADPH. Well, what do we do with it now? So then we move into something called the Calvin cycle. And so the Calvin cycle is not taking place in those thylakoid structures like the dependent reactions. Instead, it's taking place in the stroma. And does anyone remember what the stroma was?
Liquid.
Yes, that fluid-filled space that surrounds that thylakoid and the granum structures. So the ATP and the NADPH, which were generated in the thylakoid, have been transferred into that stroma and can now be used by the Calvin cycle. So again, takes place in the stroma, uses those two produced earlier, and we generate our sugar. Now I'll warn you, this next step is going to be a little heavy, but it's what we'll be repeating over the next couple days and going into more depth as we work through photosynthesis. So now we've got the Calvin cycle, and this looks kind of scary. But what you're starting with is a 3-carbon molecule, RUBP. And so this is left over from the last time the Calvin cycle ran. This is a continuous cycle. So you're going to take that 3-carbon molecule. You're taking CO2, which has been taken into the plant through the stomata in the plant leaves. And you're going to use an enzyme called rubisco. So taking those 3 starter components, you're going to enter stage 1 of the Calvin cycle. So this is the carbon fixation. And you've got your 3 carbon molecule here. You produce 6 molecules of that, 3 PGA. And the P comes from the 1-phosphate group here. And so that action requires 6 of the ATP from the light dependent reactions. So that has to run a couple times before you've got enough power to generate the Calvin cycle. And what's being done here is with that energy used, ATP is being reduced back to ADP. And that's being then transferred back to the thylakoids to be used again in the generation of more ATP. So it's constant recycling. So then we move on to stage 2, the reduction of those 3 PGA molecules. And here's where you're going to use that NADPH. So again, 6 are required here. And it's that splitting off of that hydrogen atom that was added and electrons that's going to power the next step here. So then we've got creation of 6 molecules of another carbon molecule, Ga3P. And from there, you're going to take one of those 6 molecules, and you're going to generate half a molecule of glucose. So you're only generating half from this cycle. 5 of those remaining molecules are going to be used in stage 3, which is the regeneration of this molecule that we needed at the very beginning, RUBP. That's going to require 3 more ATP, which again is going to be reduced to ADP and returned to thylakoids for more use. And so you've ended back at that original 3-carbon molecule where you started, or 3 molecules, excuse me, of that 5-carbon molecule where you started, and the cycle continues. So I know that's heavy, but again, we'll be talking and delving into this more in future lectures. But I want you all to kind of keep these 3 stages in mind for now. So what's going on? So the first stage is that carbon fixation. Second is the reduction of that 3 PGA. And the third being the regeneration of that RUBP carbon molecule. That was a lot. But let's review. Let's hit on the major points I want you to know from this lecture. So we'll do this with that Mentimeter again. So if you all want to pull back out your phones. So the order of reactions is photosystem 1 to photosystem 2, and then the Calvin cycle. Perfect. A+ students, you all did great. So again, why does 2 come before 1? Yes, I heard Rose say it. It was the order in which it was discovered, and they just wanted to make our lives difficult. So light dependent reactions occur. What's the structure that is requiring light? So you got the stroma, the thylakoid membrane, and the chloroplast membrane. Great, yes. So the stroma was that, what was the stroma? The fluid. Yes, the fluid. The thylakoid membrane. Remember that was folded over multiple times to increase surface area. And then the chloroplast membrane holds all of that together. So which stage, ignore that 2 that's on there, which stage in the Calvin cycle generates the sugar? Great, yes, so it was stage 2. That's where it's being reduced. You're using that energy from the hydrogen atom and the electrons being broken off of that NADPH. And you're resulting with a sugar being reduced here. I like whoever put that 1 dot there. That's exactly where it's happening, yes. So stage 3 is where you're regenerating that 5-carbon molecule that's going to be needed for the rest of the Calvin cycle as you continue on. Great. Well, that's great. You all know how photosynthesis works, and it works the same way in every plant, right? No, that would be too easy. We live in a very diverse world, so we've got very diverse climates and temperatures. And so plants have adapted to live in those. So the first one we're going to talk about is the C3 pathway of photosynthesis. And it's called C3 because it utilizes a 3-carbon structure. And so what's going on here? This is kind of what we've been talking about for the most part today. So you've got your CO2 that's being taken into the plant cells through those stomata openings. And then the Calvin cycle is all taking place in one cell called the mesophil cells. And then from that, you're getting your sugars. And so plants that use the C3 pathway are very efficient under cool, moist conditions where there's high CO2 availability. Scientists believe this is because they were probably the first to evolve. When CO2 was really high in our atmosphere, you have low oxygen. And the advantages of this pathway is that for each CO2 molecule that's fixed, you generate 3 ATP and 2 NADPH. And this is a lower ATP cost compared to the 2 pathways we'll talk about in a minute. The disadvantages, though, is another process called photorespiration. We'll go into more detail about this in a future lecture. But the idea behind this is that enzyme rubisco that's being used in the Calvin cycle binds to oxygen instead of the CO2. And so then more energy is having to be put into the plant to refix that carbon, get rid of that oxygen, and this wastes energy. So where you're gaining efficiency because you're producing 3 ATP and 2 NADP each year, you're losing it because of this process called photorespiration. And so roughly, depending on environmental conditions, plants are about maybe 75% efficient. You can lose about 25% efficiency if you're in a high oxygen environment. So the peanut we've been talking about today is a perfect example of a C3 plant, as well as other legumes like soybeans. But let's move on to our next one, which is C4. And so why would this be called C4 if we're following the naming scheme? 4 carbons, yep. So this is where we get into a spatial separation. So you've still got your CO2 being taken in through the stomata. You're still using that mesophyll cell that's used in C3s. But here what it's doing is it's generating that C4 from which this gets its name. The C4 carbon is then being transferred into another cell called the bundle sheath. And it's within this bundle sheath where it's being converted back to CO2. You've got your Calvin cycle operating as usual. And then the remaining carbons go back to a C3 structure, return in the mesophyll, converted back to a C4, and you have that continuation there. But now it's separated by those 2 different cell structures. So the first stable carbon compound, as I mentioned, is that 4-carbon molecule, oxaloacetate, and it's involved as an adaptation to more hot, dry environments. So you're not losing as much to that photorespiration process. Again, as I mentioned, you've got that spatial separation between those cells with the initial CO2 fixation in the Calvin cycle. And as I mentioned, the advantages of this are you're minimizing that photorespiration process, so you're not losing as much energy to rubisco binding to oxygen instead. And you've got increased efficiency under adverse conditions. So again, these have adapted to those hot, dry environments. The disadvantages, though, are that it's a higher ATP requirement initially than the C3, requiring 5 ATP and 2 NADPH compared to the 3 ATP for the C3 molecules, or excuse me, C3 plants. And so these are not as efficient under cool, moist conditions. So if you take them out of the environment for which they've been adapted, they're not as efficient. Classic example of this is corn. And so where was corn originally domesticated? Mexico. And what's the environment in Mexico? Hot and dry, yep. So you got your C4 plants. And so we talked about that spatial separation in the cells. So here's kind of an idea of what I've been talking about. So here you've got kind of your C3 plant structure. You've got your mesophyll cells here, which is where all the Calvin cycle is taking place. They still have bundle sheath cells, but they're smaller. They're not really being used for this process. And then in your C4 plant, you can see here the mesophyll cells have been arranged to surround these large bundle sheath cells so that there's that easy transfer of those carbon molecules for Calvin cycle to continue. So we've got one more pathway to discuss. These are CAM, does not follow that same naming scheme. So the C in this stands for, I'm not going to pronounce it correctly, I think Chrysalis, I think it was the succulent that this was first discovered in. A is for acid, and then M is for, I don't remember. Anyone have an idea what the M would stand for?
Metabolism.
Metabolism, thank you very much. So in this, remember in the C4 you had that spatial separation. This you're going to have a temporal separation. So you've got your CO2 being taken into the stomata, but if you notice it's being done at night. And so it's taken in those C4 carbons being stored or taken in by the mesophyll cells. And then during the day, that's being converted back to CO2, being used in the Calvin cycle, and you've got your sugars as your final product. So again, you've got that temporal separation between the two processes. These again are adapted for your dry, arid, desert-like conditions. The advantages of this is because it's got excellent water use efficiency. So if you think about the desert, you think if we go out there, we sweat a lot, we get dehydrated when we're in the sun. Plants are no different. So if they've got their stomata open during the day, they're gonna be losing a lot of water, right? So if you close them during the day and you open them at night to take in that CO2, you're not gonna lose as much water. The disadvantage of this, though, is that it's got a slightly higher energy cost compared to your C4 plants. Because when you're taking in that CO2 at night, and then you're having to wait for that sunlight energy during the day, you're having to store these molecules, you're having to transport them. So there's some energy costs involved with that. And then you've got slower growth due to the limited CO2 fixation. So since it's only taking CO2 in at night, you can only do as much during the day as what you're taking in. So here's kind of another example or an idea of what's going on. So you've got your CO2 coming into the plant at night. It's being transformed to that oxaloacetate, that C4. It's then transformed into a malate and then a malic acid. And so it's this malic acid that's being stored in the vacuole overnight for use during the day. So then we come to the day, that malic acid that's been stored in the vacuole is then transported out, back to malate, back to CO2, and then the Calvin cycle can continue, as we discussed before. So again, there you've got your light during the day, you've got your stomatus closed. Pineapple is a great example of this. Interestingly, I've heard people say that if you harvest and eat pineapple at night, it's much more acidic. And why would that be? You're storing that malic acid, yep. And then during the day, it's much sweeter because that malic acid is being transformed back into malate, back into CO2 for energy production. Okay, so we've talked about the three pathways. What was the pathway that's evolved for cool, temperate climates? C3, I see it in the back, thank you. The one for more arid, hot, or excuse me, yeah, more hot, dry environments. C4. And then the one for your deserts, CAM. So today we've learned the chemical reaction of photosynthesis. We've talked about the light-dependent and independent reactions, and we'll dig deeper into those in the next couple lectures. And we've talked about the three pathways for photosynthesis. Okay, cool science, but where's the real world payoff? Why do you guys need to know what photosynthesis is? Why is it important for us to understand this? So we're going back to our peanut plant. So our one little peanut plant produced the peanuts that you enjoy, but it's just one plant. But that one plant is a bigger peanut field. This is a peanut field at one of our research stations in Virginia. And then that one peanut field is part of a whole ecosystem. And so we've got this delicate structure of plants that are generating the oxygen as their waste product for us to breathe, and sugar for their energy and ours. And then we're breathing out and providing CO2 to them. So that's just kind of a very basic idea of how we're benefiting from this, why it's important to understand it. So I want us to spend some time together today and brainstorm on why you think it's important. How can our understanding of the photosynthesis process be used in our world today? So I'll start you off just kind of with a topic to start with. How can we use our understanding of photosynthesis to maybe increase food production? Crop management, yes. So what would be important about that? Yes, exactly. Yep,
Leaf area leaf management.
perfect, yes. So you want to talk about how you're managing your cropping systems, because if you plant them too close together, you're limiting the amount of leaf exposure to sunlight. If you're working with a C3 crop, you're increasing the amount of oxygen that's surrounding those, so you're gonna have more photorespiration, less energy output, so less yield. What if we think about gene editing? How can we use gene editing to increase food supply? So which of the pathways was more efficient? That here's C4, yes. So do you all think we could adapt a C3 plant with gene editing to C4 and increase its efficiency? Yes, yep. There's some research going on with that. I don't know of any successful varieties currently, but I know there's been a lot of work with rice. So if you increase the energy of efficient of a plant, where is that energy? How are they going to use that energy?
Increase yield.
Increase yield, that's the hope. We're hoping it's going to increase yield and not just vegetative growth, right? So we can use that. Any other ideas from anyone before we go on? Great. So here are just some publication titles, exactly what we've talked about. Photosynthesis and security, how we can, the evolving story of C4 rice, how they're hoping to adapt rice, which is a traditional C3, into a C4. Studies, again, on planting methods and the effect of high planting densities on photosynthesis and so yield. And then you can kind of get more into the engineering aspect and medical advancements. So there are medical researchers out there who are using nano-enabled photosynthesis in tumors to activate lipid peroxidation for overcoming cancer resistances. Big title. Anyone have any idea what that's referring to? So they're creating these little nano-enabled particles, and they're modeling it after photosynthesis. So it's taking in CO2, it's releasing oxygen. Tumors don't like oxygen. That oxygen is being delivered right onto the tumor site. And that's all based on their understanding of photosynthesis. And our last, green energy. So solar panels do okay. We can definitely improve them. And so there's research being done to model solar panels after artificial photosynthesis. And so powering a Green New Deal for sustainable energy. So you don't have to just be going into plant science in order to have an appreciation or need an understanding of photosynthesis. There's so much that's still being done with it. And with that, I'd love to take any questions. You don't have to participate in this through Mentimeter. I like offering this at the end of the class for any students who are more shy or less comfortable asking in front of the audience. And so I'm happy to take questions either way. Is it open? Are people able to access it? Yes.
There was a lot of detail in those reactions. Do we need to know all that detail for the exam?
We're going to be going into it in future lectures. I'm going to make sure everyone's comfortable with it. Everything I discuss in class is up potentially for the exam. If I don't mention it in class, it's not going to be on the exam. We're going to get everyone comfortable with it. Yes.
Dr. Fletcher, this is just so much. And I was wondering if there was going to be like a study guide or something for the class, maybe. I know our first exam is coming up soon.
Yes, I'll provide kind of a structured study guide. I won't give exact answers to anything, but it'll be kind of a guide to help you to know what to do. I'm also happy for you to come visit me during office hours and we can go through this in detail.
We participated in the Menti questions. Do we get bonus?
So I do keep these as a record of participation. I'm not necessarily taking attendance, but if we come to the end of the semester, you've come to class every day, so I'm happy to work with you all. Okay, so elaborate on the dependency of the dark reaction on the light reaction. Okay, so what I want you to know from today's takeaway is that what's being generated in the light reaction are those two ATP, NADPH being generated, and that's what you need to power that dark cycle reaction. So without the light, you don't have that energy for the Calvin or the dark reaction. So that's what I want you to take away from today.
Raise hand.
How do I do that on the Zoom computer?
You can press the date one on the keyboard and then
Okay.
it'll bring up the chat. Perfect.
Oh, that was me.
Okay.
Okay,
Somebody had their hand up.
Yeah, it was.
Jumping.
Jonathan, you can unmute yourself and just ask.
Yeah, very nice lecture, and I'm wondering, do you have any practice questions or some homework questions we can work on to help us to understand the materials? It's just too much detail and a lot of to study. If you can provide some practice questions, that would be helpful, I think.
yes, I'm certainly happy to provide extra practice questions. Again, all of the Mentimeter questions, I'm gonna provide in a folder for you all to use as review material. And so the Mentimeter questions that I ask in class are a great guide for your exam questions and the material I want you to know. Does that help?
Thank you.
Yes.
Are there implications of climate change on the efficiencies of the various photosynthetic pathways?
Yeah, so that's something that's certainly being studied. It's certainly one of the reasons people are so focused on converting C3 to C4. So there's that increased efficiency and adaptation for those hotter climates, which we're all quickly becoming accustomed to, hopefully. And one of the concerns too is with C3 crops, while it's more efficient at, it uses more, well, the thought is that it would use more CO2 if it's in the air, but there seems to be a limitation on how much a plant can take in. So C3 crops probably won't be our answer. C4 seems to be the way most researchers are thinking at the moment. And we can continue to adapt through gene editing, hopefully continue to produce the food necessary through climate change. Yes.
So since you mentioned that a couple of times, I'm not clear how you will use gene editing to change your C3 to C4. How would you do that?
Yes, so I'll just briefly touch on it since this isn't a gene editing class or a breeding class. I'm happy to go into more detail with you outside of class. But for those who are interested, you would adapt it so that there's more structure in the mesophyll cells. You would take the genes that are being expressed in those C4 plants and hopefully insert them and have that expression in your C3.
But would that be transgenic or would that be gene editing?
Depends on what method you use. So CRISPR, I think, is legally classified now as non-GMO. But if you're going to use a classic method like agrobacterium, that would be considered GMO. And again, I'm happy to, more than happy with my background to answer those questions outside of class.
Yeah, but now I'm really confused. To me, gene editing is you make a knockout or you change some nucleotides, right? But you're not putting in a new gene, right? Is that correct?
Well, again, I'm happy to elaborate. I don't want to confuse anyone else in the class. This will not be on the test. So if you want to come to my office hours, I'm happy to discuss all of that with you.
Okay.
Yes.
Do all the plants fit into one of these three categories, like C3, C4, or CAM?
That would be too easy. So these are the three major pathways. There are some plants that seem to be kind of caught in that evolutionary loop where they're like between C3 and C4. And it's really kind of an interesting real-time study on evolution. But these are the three you need to know. I won't ask for anything else, but yes, there are some that fall outside of these categories. Anything else from online? Great. Anything else from the live audience? Okay, thank you all very much.
So certainly others are welcome to stay. You know, we're gonna have an opportunity for faculty who have not met with Bodie, or even those who have and have shared a meal to just, you know, have conversation, opportunities for you to ask us questions. So this is your time to sort of meet with faculty, but you know, if you're here and you're interested, I know there's a lot of graduate students here. If this is a convenient time, we didn't have as many students, you're certainly welcome to stay for a little while or ask your questions in the beginning. I don't wanna take, you know, away an opportunity if you're interested in talking to the candidate, you know.